So good morning, students. हम लोग today we will talk about micropaleontology. Moreover, in the offline mode, we studied the Unit 1 and we covered a major part of Unit 2 as well. So in this Unit 2, let's begin with the revision of Unit 2, that is Foraminifera. In this lecture, we are going to study about the habitat, the test morphology, the ultrastructure, and everything related to the morphology of Foraminifera. So let's begin today's lecture. I'll share the slide. Is it visible? Mehul?
Yes, sir. It's visible.
Okay. So this lecture will cover actually 3 of my online lecture, which is the previous lecture, the today's lecture, and tomorrow's lecture. So this is the slide which I'll share at the MLSU website as well. So as we already know about micropaleontology, that in micropaleontology we generally deal with all the organisms which can only be studied under the microscope. Okay, so when we say micropaleontology, it doesn't mean that we cannot see it by naked eyes. We can see it through naked eyes, but we cannot identify or name those creatures. Okay, so for taxonomic purpose, we need to have this. Somebody's trying to join. Just let me add him. Please join. Problem in online class. So when we say micropaleontology, it simply means that the organism can be seen through naked eyes, can be visible through naked eyes, but it cannot be studied without the aid of microscope. So those organisms which can only be identified at the taxonomic level through microscope comes under the purview of micropaleontology. So if we say micropaleontology, so it includes various organisms like Foraminifera, nanofossils. Nanofossils doesn't mean that 10 to the power 9. Okay, it's still in a micron meter range. It is called coccolithophore. Okay, so they comes under the purview of a nanofossil. Then we have ostracods, pteropod, Dinoflagellate cyst, and etc. So these organisms, they are unicellular to multicellular. So when we say micropaleontology, it includes unicellular organism as well as unicellular as well as multicellular organism. So if somebody asks you Foraminifera is unicellular or multicellular, then you will answer that it is a multicellular. But other microorganisms like ostracods, they are multicellular. Okay, so in Foraminifera, these are typically unicellular organisms. Okay. Also, sometimes the skeletal remains of a larger organism, which are very minute in size, they are also considered to be as part of micropaleontology. Okay, like conodonts. We don't know about the affinity of conodont. Affinity means with which organism it is related to. So conodonts, which are typically phosphatic microfossils, these microfossils are very tiny in shape but have varied structures or shape. Till today, we just understand about the taxonomic concept of the conodont species, but the actual affinity of the creature, how the organism look like, how they move, or about their other physical characteristic, we don't know much about it. It is all assumed through various studies. So again, when we call conodonts, it's the creature that leaves the remains of conodont teeth or whatever the shape is. So the conodont organism is typically a lizard-shaped organism that we guess. Okay, but still these conodonts are the teeth or the dentitions of those lizard type organisms. We don't actually know about the organism, but we guess from these conodont structure that this organism must be like a lizard. Okay, so just in the case of conodont, as the organism is very large, but the remains of that organism or the dental remains of the organism is microscopic in size, so we consider those dentition pattern as a part of micropaleontology. So is it clear? Mehul, Bhuvnesh, is it clear?
Yes, sir.
So whenever you have any doubt, you can just ask me in between. Okay, you don't have to wait till the end.
Yes, sir.
So the objective of micropaleontology is all about studying their morphology, their structure, their chemical composition, and their mineralogical component as well. Because the mineralogical component of the organism is very much important for understanding the water chemistry or the ecological environment in which they live. Okay, because the composition of their test or shell, they typically depends upon the composition, water chemistry of that environmental niche. Okay. Also, for micropaleontology, the morphology is very important. Morphology is important because of the species concept in paleontology. The species concept in paleontology is typically a morphospecies concept. Morphospecies means those organisms having similar morphology as considered to be a same species. So similar looking morphology or same morphology is considered to be a single species. Okay, so we need to study the morphology very carefully in order to understand their species concept and distribution in the morphospace. Also, the structure means the test structure. When we take a cross section of the test, when we cut the text in a Y section, you will find that the test is composed of various layers like these fingers. So based on these layering, we can also try to classify those microfossils as well. Okay. Then other part is about the to discover the origin and systematic affinities. So in order to understand micropaleontology and in order to classify these microfossils, we need to understand from where they evolved, how they originated. So for this, we need to apply the law of faunal succession. The law of faunal succession is about the complexity of life increases with time. Okay, but these are unicellular organisms. You cannot apply it there. So through biostratigraphy, you just understand which organism are occurring at particular origin. So suppose some organism appears at 5-meter depth. Okay, so it means that it must have evolved from a species which are present below 5-meter. So their parent must be in this below 5-meter region. So we need to look at the morphology of the ancestor and descendant in order to analyze their relationship, their phylogenetic relationship. Also there is another way to understand the origin, that is through ontogeny recapitulates phylogeny. So what happens in ontogeny recapitulates phylogeny? Can anybody tell me? What do you mean by ontogeny recapitulates phylogeny? Bhuvnesh, समझ में आता है?
No, sir.
Ontogeny recapitulates phylogeny का मतलब क्या होता है?
No, sir. नहीं है.
Mehul? Online class.
Idea.
हाँ. Ontogeny recapitulates phylogeny का मतलब क्या होता है? देखो, ontogenic growth का मतलब होता है, ontogenic growth means the development stage from a child to a young individual. Okay, ontogenic development. Okay, so ontogenic development में क्या तुम देखते हो? तुम just imagine your childhood. When you take birth, what you are doing? You're just sitting on the couch. Then you start crawling. Then you start kind of reptile kind of movement. Then you start standing erect. Earlier, you were trying to crawl on 4 feet. Then you try to take a straight position, and then you start walking. Okay, so this is called the ontogenic development, from younger to elder stage. The development which occurs from the juvenile to a mature stage. This is called ontogenic development. So this ontogenic development actually represents your phylogenetic evolution. Phylogenetic means from where you evolved. We all evolved from unicellular organism. But we trace our ancestry, then we are all mammals, and mammals evolved during Cenozoic. We don't have mammals in Mesozoic. So when we go to Mesozoic, what we find? There are birds, there are reptiles. Then we go to Paleozoic, there are fishes and amphibians. So fish, amphibian, reptile, birds, and then mammals. So this is phylogenetic growth. Do you understand? This is phylogenetic growth. So phylogenetic ग्रोथ में आपको ये phylogenetic जो आपका evolution है, you can also study within an individual from its ontogenic development. Okay, so if you analyze the embryo of a fish, of a reptile, of a mammal or other organism, you find that the embryo of all these individuals are same. Okay, it means that they all evolved from a same ancestor. Okay, and later on they acquired the different ecological niche and they have totally different morphology. Okay, so now is it clear? Now is it clear? Bhuvnesh, Mehul?
Yes, sir.
Ontogeny
Yes, sir.
recapitulates phylogeny?
Yes, sir.
Yes, sir.
Good. So to understand the evolution and origin of micropaleontology, we typically don't go for law of faunal succession. We go for ontogeny, recapitulates phylogeny, or biostratigraphy, depending upon where the organism is occurring in the geological section. Then what is the application? We will study about the application of these micropaleontology in oil exploration, the biostratigraphy again I discussed, biopaleology and paleoclimatology. These are the important application of important field in which the micropaleontology is applied to a very much extent, and we will study it. And then what interference we made about their environment. So first we will start with the microfossils group, as this is the first video recorded lecture. So microfossil groups, as I just mentioned earlier, we have Foraminifera, which is typically unicellular. So when we say Foraminifera, you have to understand that it is a typical calcareous, CaCO3. Okay, don't confuse it with aragonite or anything else. Foraminifera means calcium carbonate, means calcareous. Okay, then it is typical marine. Okay, it is a typical marine organism. It lives only in seawater, nowhere else. Okay, but just to mention here that there is a single species of Foraminifera which is totally a freshwater species, but this is not included in this chapter, and that is not much studied as well. So if somebody asks you what is Foraminifera, you must say it is a unicellular organism, it is of calcareous composition, and it is a typical marine organism. Okay, then there comes a word called agglutinated. Agglutinated. Now, what do you mean by agglutinated? कौन बताएगा? Agglutinated का मतलब क्या होता है?
Not glutenated.
And glutinated का क्या मतलब होता है? सही जा रहे हो मेहुल? Just try. भुवनेश?
जी सर।
बताओ, एग्लूटिनेटेड का मतलब क्या होता है? मेहुल तुम गेस कर सकते हो, डोंट वरी। ग्लूटिनेटेड का मतलब होता है इकट्ठा होना, चिपकना, ठीक है। तो उससे वर्ड आया एग्लूटिनेटेड। एग्लूटिनेटेड का मतलब होता है आसपास की चीजों को इकट्ठा करके चिपका लेना, उसको बोलते हैं एग्लूटिनेटेड, ठीक है। एग्लूटिनेटेड का मतलब होता है चिपका करके अपना टेस्ट बनाना। मींस मैं जहां पे रहता हूं वहां से, जैसे अघोरी बाबा होते हैं ना, वो भस्म लगा लेते हैं और नंगे रहते हैं, जो शमशान में रहते हैं, उसी तरीके से ये जो कुछ फोरामिनिफेरा हैं, कुछ, नॉट ऑल, few foraminifera, what they do is they collect minerals or whatever material is lying on the sea bottom and they stick to their shell. Okay, so typical उनका कोई composition नहीं होगा, it all depends upon in which substrate it is living. So जो भी उनके आसपास में होगा वो अपने शरीर में उसको चिपका लेते हैं, and they are known as agglutinated forms. Okay, marine, they are planktic as well as benthic, planktonic and benthonic. So planktonic means those who live at the surface water, and benthonic means those who live at the bottom, sea bottom. Sea bottom means where the bottom and sediment water interface is lying. So benthonic organism, they live at the sea water, sediment water interface. Okay, and then comes the heterotrophic. Now what do you mean by heterotrophic, Bhuvanesh? Bhuvanesh, Mehul, what do you mean by heterotrophic?
Sir, I guess water loving.
No.
No, auto,
Oh,
autotropic and heterotropic दो terms होती है सर.
हाँ,
I guess.
ऑटोट्रॉपिक क्या होता है, हेटेरोट्रॉपिक क्या होता है? गुड, यू हर्ड अबाउट ऑटोट्रॉपिक। देखो, ऑटोट्रॉपिक का
सर वो
मतलब
खुद फूड जो.
एग्जैक्टली, वेरी गुड, जो who make their own food, like photosynthetic organism, those organism who are capable of making their own food, like plants. Okay, so they are called autotrophs. Now those organisms we cannot make their own food, they are called heterotrophs. Okay,
Heterotroph.
so those organisms which cannot make their own food, they are considered as heterotrophic organisms. So in case of Foraminifera, they are incapable of cooking or making their own food. Okay, understand?
Yes sir.
Okay, good. So Foraminifera are unicellular, calcareous, agglutinated, marine organism, planktic to benthic, and heterotrophic. So these are the 5 or 6 terms you should remember, and you just write a sentence over it when somebody asks you about Foraminifera. Okay. Now coming to the Ostracoda. This is totally about the introduction, and we will move into the details in the coming lectures. So Ostracoda is about multicellular organism. They are calcareous as well as chitinous. Now, chitinous is totally different composition, and in ostracod we call the test or the outer covering as walls. Okay, so there is right wall, left wall. They are marine as well as freshwater. Okay, so they are found in continental or terrestrial to marine environment. Okay, they are found in both the region, marine and terrestrial. In terrestrial we have freshwater, and in marine, we have salty water, and we have different kind of ostracods. Okay, so you just understand it like it's not just one organism. Like humans, we are Homo sapiens, but we have different morphology. The American look different, the Japanese look different, the Nigerian look different, the Indian look different. Even in India, the Northeast people look different. So the appearance of organism, it depends upon in which habitat they live. Okay, so we don't call a Nigerian a different species. We call it a Homo sapiens. Okay, so likewise, the ostracod also have different n number of species living on different ecological niche. Okay, they are also planktonic and benthic, like living on the surface and living in the sediment water interface, and they are also heterotrophic. So they also depend upon some other organism for their food. They don't make their own food. They have to depend on somebody else to cook their food, to make the food for these organisms, and then they take the meal from them. Then Coccolithophora. Now Coccolithophora is called also known as Nanofossil. Nanofossil. Okay, they are very minute, 10 to 20 micron meter range. So they are also they are just like foraminifera. They are unicellular, but they are autotrophs. They make their own food. So you can see the contrasting character of different microfossil group. Ostracoda, heterotroph, Foraminifera, heterotroph, but Coccolithophora are autotroph. They are typical calcareous, no agglutinated, nothing. Okay? They are planktonic and benthonic, and they live in marine and fresh environment. Okay? Then we have diatoms. Diatoms is unicellular autotrophs, but they are siliceous. And they are not benthonic. They are typically planktic. Okay? And they are mainly marine, but you can have diatom in freshwater as well. Okay? So that's why it is mentioned that mainly marine. Okay? So diatoms can be used for analyzing so many things. Even for a lake also, you can go and study the diatom composition, diatom assemblies, and you can ascertain whether the lake is productive or the lake is contaminated by different heavy minerals as well. So different, if you change the water chemistry of a certain water mass, the diatom, they evolve, or the different assemblage of diatom species will appear in the ecological niche. Okay, so for diatom, they are mainly marine, but they are found in other water as well. Radiolarian, they are typically marine. They are again unicellular, heterotrophic, siliceous, and plankton. Now comes the dinoflagellate cyst. Okay, so dinoflagellate cyst compositions, there are typically different varieties of dinoflagellate cyst. They are of organic walled microfossils. So these dinoflagellate cysts are also typically marine and freshwater, and they are autotroph and heterotroph as well. So we will discuss this in detail about the dinoflagellates in Unit 5. Okay. Is this figure visible? Is this figure visible to you all?
Sir, which one, sir? Which one, sir?
All. So this, you can see my cursor? Cursor दिख रहा है?
जी सर. Yes sir.
कर्सर में आप देख
Yes,
रहे
sir.
हो, ये कर्सर में आपको जितने भी फॉर्म्स दिख रहे हैं, ये बेंथिक फोरामिनिफेरा के फॉर्म हैं. They are all बेंथिक फोरामिनिफेरा. ये देखिए, ये स्पाइंस है, ये छोटे-छोटे डॉट्स हैं, ये बेंथिक फोरामिनिफेरा. यहाँ जो दिख रहा है आपको, ये सारा आपको डायटम्स है. ठीक है? यहाँ रेडियोलेरिया है, ऑस्ट्राकॉड है, ये कोकोलीथोरा, ये जो है ये डाइनोफ्लैजेलेट सिस्ट है, और ये टेरोपॉड. Just for a glimpse that how their outer morphology is like, I'm showing it. Okay, just for a sake of convenience. Okay, so just look at these. So when you see these organisms, what comes to your mind? How can you differentiate between pteropod and benthonic foraminifera? Can you see? Can you see any difference in pteropod and benthic foraminifera or diatoms? You can see the difference. Yeah, yes. You can see the difference in the shape. Okay, so we will have these benthic foraminifera and pteropods in our lab as well. Mehul, I think you have done some practical as well. Have you seen these pteropods?
Yes, sir.
Have you seen the pteropod?
Yes, sir.
I think you have seen this. Okay. Let's move to the unit 2, which consists of foraminifera in detail, and we'll try to cover this topic. So when we say foraminifera, so foraminifera, as I said, it is a single cell organism composed of a test, which we call shell as well. There is a shell, there is a test. So if somebody asks you the difference between skeleton and shell, what you will say? What is the difference between skeleton and shell?
Sir, shell is the outer covering.
Very good.
While skeleton is, sir, skeleton
A skeleton
Means
provides the inner strength of muscles, or it is just like endoskeleton and exoskeleton. Endoskeleton is inside the body to give the shape or strength to muscles, or the structure to the and give shape to the body, while exoskeleton protect the body from outside. Okay, so it is just like exoskeleton, it covers the body from outside. Cell is the outer covering. Okay, it don't go penetrate within the body. Okay, and the foraminifera are made up of different materials, like they are chitinous, calcareous, agglutinated, and siliceous. So if somebody asks you, now remember this, it will appear in many exams as well. If somebody asks you the composition of foraminifera, okay, the composition of foraminifera, okay, so you have to look at the options. What are the options? Okay. So then again, you have to look at the question very carefully. If it is asking about foraminifera, then you will say all of the above. It is saying planktic foraminifera, then you will say calcareous. For planktic foraminifera,
please pardon, sir.
bolo.
Please pardon, sir, what you said.
Okay. So
repeat.
in exams, there are several questions which appears like, what is the composition of foraminifera? Okay. So in that case, you have to look at the options, the answer options. So what you will see, if it is they are asking what is the composition of foraminifera, and you will see chitinous, calcareous, agglutinated, siliceous, then you will mark all of the above. But if the question is asking about what is the composition of planktic foraminifera, then you will mention only calcareous composition. Okay. Okay. So calcareous composition होगा planktic foraminifera का, और बाकी के केस कुछ भी इकट्ठा करके अपने बॉडी पे चिपका सकता है. Okay. And if somebody asks you about the range of these microfossils, typically in foraminifera, the ranges from 0.1 micrometer to 1 or 2 micrometer. Even in some cases, they grew up to 10 centimeter as well. Okay. Have you heard about Fuselini's, Melolitic limestone? These are larger benthic foraminifera. Okay, larger benthic foraminifera. Have you been to Akli Formation, Giral lignite one? Giral गए हो?
Yeah, yes sir.
गए हो? वहां पे देखा है तुमने benthic foraminifera का? नहीं देखा. तो जहां भी तुम पेलियोसिन में जाओगे, पेलियोसिन इयोसिन एज के रॉक्स में जाओगे, वहां से ये मिलेंगे. ओके. Now the animal of these foraminifera, अगर हम as an organism की बात करें, as a micropaleontologist or paleontologist, what we study is the outer test. Okay, हम सिर्फ बाहरी जो संरचना है, बाहरी जो टेस्ट है, उसकी स्टडी करते हैं. But now the paleontology or the micropaleontology is also evolving with time. Okay, now the period or the era is of molecular paleontology, in which we study the RNA, the DNA of these fossils, and we try to just establish the ancestor-descendant relation, not typically on the morphology, but on the DNA makeup of the organism. Okay, so in present scenario, okay, so department are closed. Just got a message. Okay, so what will happen? What I was saying, do you remember what I was discussing?
Sir, you were saying that now we are studying the DNA and RNA of the microphages.
Exactly.
Sir, is it still the presence is still there of DNA and after thousands of million years?
No, not for thousands of millions years, but just for a organism of up to 10 to 20 million years, it is possible to extract DNA from those organisms. It's possible. It's possible for 10 to 20, not beyond. Okay, because what happens with time, the DNA structure, they degraded with time. Okay, so that totally changes the data. Okay, but there are few micropaleontologists who are still trying to study these DNA structures. There are a few in Pune, in Agarkar Research Institute. And also there are a few recent papers by this group related to the differences in the species. Like earlier, as a paleontologist, we consider sinistral and dextral. You have also heard about these terms like sinistral species and dextral species. Have you heard
Yes,
about
sir.
it?
Yes, sir.
Okay. So in micropaleontology also there are species which are considered as sinistral and dextral. So what paleontologists suggest was like the sinistral population was dominated during the cooling interval, and the dextral population is dominated during the warm interval. So this sinistral and dextral, they used to think that the organism changed their cooling direction due to temperature or something. So the organism is one, but it changes its coiling direction, the manner in which it adds the chamber to the ship. Okay, but later on, with the development of this molecular paleontology, the micropaleontology or the paleontologist community, they understand that these dextral and sinistral, these are two different species. They are two different organisms. Typically, the species are totally different. They are not sinistral and dextral variant. They are totally different species. Okay, so through DNA, we come to the conclusion that these two are totally different. So people are still trying to understand these species concept in paleontology right now. So if we start discussing about the organism, which we are very less care about as a paleontologist or a geologist. We don't care about the organism, what is the Golgi apparatus or what is the ribosome and mitochondria as well. But as a paleontologist, we try to look at the outer covering of the cell structure. Okay, but right now in present condition, with the advances in paleontology, for for doing some impactful research, you need to understand the fundamentals as well. So for this, we need to understand here what is the composition of the organism of Foraminifera. So it consists of protoplasm. We all know about protoplasm. Protoplasm covers the nucleus. It is the live fluid in which all the cell organelles live. Okay, so protoplasm is the organism in which there is a nucleus. Now, this is if somebody asks you what is the organism, you will say this is the protoplasm. Okay, so protoplasm is surrounded by membrane. Now, protoplasm are also called endoplasm. Okay, so this protoplasm, so we will have in cell what we will have protoplasm, then we will have nuclei or nucleus. When we have one, we will call it nucleus. When we have more than one, we will call it nuclei. Now, then we have a cell segregation. Now, there are different organs like we have liver, kidney, bladder, small intestine, lungs, heart. Likewise, the cell also have these Golgi apparatus. Okay, then they have mitochondria. Okay, mitochondria, the powerhouse of the cell from which it extracts energy. Then they have
Energy.
Then they have something called ribosomes for protein synthesis. Then they have vacuoles in which they just fill the gas and they try to assimilate the foods. Now, there is a protoplasm that is outside the test. So the protoplasm which is inside the test, and there is a part of protoplasm which is outside the cell. So you have seen the test under the microscope in the practicals, and I told you that there are several pores or the several openings in the test. So through these openings, the protoplasm, the organism which lives inside, they extend their body to the surrounding environment. So what the part of organism which remain within the cell, they are called endoplasm, and the part of organism which are extended outside the test, they are called ectoplasm. Okay, and they form the structure called pseudopodia. Pseudopodia, you have heard about it when you studied this amoeba in your school. So pseudopodia are the irregular shape of extension from the body, which is generally used for capturing or gathering the food and for feeding purpose. Okay, so these pseudopodia, they have typical length of 2 to 3 times or even 20 times the test diameter. So the test, suppose test is 1 micrometer or the 1 millimeter, then the length of the pseudopodia might goes up to 20 millimeter. So it is much more than the body itself, like our hand. The length, the width of our chest is 32, 34, but the extension it must be covering almost 2 meters. Okay. Likewise, the pseudopodia, the extension, it increases the extension of the organism. Okay, they can branch very often, shaping web-like appearance around the test, surrounded by rather fluid layers. So these pseudopodia, these are in different shapes. Like they can be single strings, they can be branching, they can be web-like pattern. There are different kinds, and they just float and they just move. Okay, to gather the food. Now, what are the function? Function is like they are like just hand. They try to feel the object. Changes in the chemistry of the environment, they are used for food gathering and transport. Now, food particles, they are caught and incorporated in the ectoplasm, transported of food through endoplasm through apertures. Now you can see the beauty of these organisms. Through pseudopodia, they collect the food. Okay, they collect the food. So ectoplasm, through ectoplasm, they collect the food, but they don't take the food through ectoplasm and transport it to the endoplasm. They take the food and take it to the mouth, which is aperture. Now, through aperture, they transport the food to the endoplasm. So, and also it is used for waste product outward. So, waste को भी बाहर जाने के लिए ये इसी तरह use करते हैं. Now, the benthic fixation and motion, now how they move or they are there. So, benthic forms, they are sessile and motile. Again, you understand. This line is very important. If somebody asks you, benthic is sessile or motile, so that might create a confusion. Because planktic is floating on the surface, it doesn't mean that it is swimming. It is swimming or they are swimmers. Okay. Planktic remains in the surface, but they cannot move on their own. Okay, they are non-motile. They are passive dwellers. But benthic organism, they are motile. Okay, they can crawl and they can move. Okay, so you must remember this. Planktics for Foraminifera migrate without pseudopodia action. They can move only by the effect of water currents or gas volume variation. Planktic Foraminifera, they migrate without pseudopodia through water current. So, ये इनका pseudopodia नहीं भी रहेगा तब ये move करते हैं through water current. ये खुद move नहीं करते, इनको water current एक जगह से दूसरी जगह ले जाता है. Just like dust. हवा dust खुद transport नहीं होता. They need a medium for transportation. Likewise, these planktic foraminifera, they need a medium like water, or they need a effective transporter. Okay. Now, formations of new chambers. Now, these.
Sir, 1 second. Sir, 1 second. The one which can move on their own are called as motile.
Yes.
Okay, sir. Okay.
Those which can migrate, they are called motile. They are called active dwellers. Okay, active dwellers. And planktic foraminifera are called passive dwellers.
Passive.
Okay. Now these are few photographs of foraminifera. Now you can see these extensions. This is the organism, and these are the extensions, pseudopodia. Okay. In the last class, I told you about the reticulogranulose texture. Now, if you look at these pseudopodia, you can find tiny granules over it. Okay. So these are branching pseudopodia. There's web-like pattern. So these are different morphology of foraminifera. Now coming to the life
Sir,
story. Bolo?
once you can you please shift to the previous slide, sir, for 1 minute?
Yeah.
1 second. Okay, sir. Thank you.
Okay, so I think it's already 11:50, and I think the online class is enough for 40 to 30 minutes. So you can now discuss. We can study about other things in the next class. Okay, in which we will discuss. We will try to complete this topic. Okay. What I will do is I will upload the video. Okay, I will try to upload the video of today's lecture as well as the PPT on the MLSU website. Okay, and share the link in your WhatsApp group. Okay, so if you have any doubt, you can just ask. If you have any query, any doubt. Mehul Bhuvnesh.
No, sir, I guess
So
we will revise and
how's the experience of Zoom app and Google Meet?
Zoom app is much perfect, sir.
Yes, sir.
भुवनेश ठीक से आ रहा है?
Zoom. Yes, sir. Zoom better.
Zoom better है. चलो.
Yeah.